Good morning, everyone, and a warm welcome to each one of you. I'm Dr. Gaurav Agnihotri. I'm Senior Manager Regulatory Affairs working at Kenview, and I'm a local executive committee member of AFSTI Mumbai chapter. As we know that we have got a very fantastic session lined up for today. So we welcome you, Dr. Shruti, for the same. Uh, I have been given this responsibility to introduce you and brief the AFSTA and CSAMB as well. So I'll be doing that, and post that I'll hand it over to you, Dr. Shruti, so that we can take it forward from there. I'll be sharing my screen because we have got few slides for you all to know what exactly AFSTI and what we do. So let me give a minute's time. I'm bringing that up. I hope my screen is visible.
Yes.
Yeah, thank you. So as we all know that today's session of Food Tech Sherpa is being organized by AFSTI Mumbai chapter and CSSMB. With this, I'll like to introduce the AFSTI, which is nothing but the Association of Food Scientists and Technologists, which is one of the largest organization which is working in the interest of food professionals. And that's how this is being introduced to this country, which is having its headquarter at Mysore. And Mumbai chapter is its second oldest chapter because it has got 40 plus different chapters spread across India. So that's how the AFSTI uh, is spreaded across this country. The main part of AFSTI is uh, it it works on the advancement of knowledge base of various aspects of food and technology, and it also aims to promote rational and economic development of food science and technology in this space. It has been holding many symposia, many conferences, not only nationally but also internationally, in the same spirit of bringing food and food technology and engineering to the next level. We also know that AFSTI is also keen towards a couple of policy shaping activities. That's where we work with government agencies on couple of ongoing agendas wherein industry also comes forward. We put forward couple of proposals, and that's how AFSTI also works towards bringing the policies to the next level. So that's primarily what AFSTI is being doing. Now, as we know that AFSTI has its important levers connected to regulators, academia, students, and industries. That's where we encourage a lot of students to come forward and be a part of AFSTI, because it will not only help you guys build your networking, but as a part of ongoing and continual process of mentorship and academia guidance, couple of industry projects or few of those research and development projects which are ongoing, those can also be taken up to the next level is what we believe here. That's where we, through this educational series, we are bringing our eminent speakers to you. We are helping you guys with couple of new advanced topics for the betterment of understanding this food technology in another way. And that's where we welcome and we request and urge you come forward and please be part of AFSTI's journey. Now, as we know that there are many initiatives that AFSTI has been driving, and few of them just to touch upon is something like IFCOST, which is nothing but the convention of food scientists and technologists, which this team has been organizing, and we have lined up one such IFCOST meeting in the month of December. It is organized on 19th of December, and it will last till 21st of December. That's where we also encourage you guys to be a part of this association, wherein you can gain and upskill your understanding in the food technology space. Now, for today's session, CASMB is also our partner. And as we know that Chamber for Advancement of Small and Medium Business, they work with their stakeholders uh, for the advancement of micro, small, and medium business to make them more resilient and enable them to introduce to the right market so that they can bring right products to the market at the right time. So that's how CSMB has been doing in this space. So we also thank Chamber for their partnership. With this, I'll now go to introduction of our today's speaker, Dr. Shruti. Dr. Shruti, first of all, welcome. And you know, for the benefit of our participants today, I'll try to give justice to the profile. I know it's a very interesting profile that I have been going through. So Dr. Shruti is assistant professor in Department of Life Sciences at SK Somaiya College. She's currently working in this department, and her academic journey has begun with strong foundation in biotechnology only. So she has completed her BSc and MSc from VG Vazay College and earned PhD from a very prestigious and reputed institute, Institute of Chemical Technology, Mumbai. She is also principal investigator of a noteworthy project titled Valorization of Biowaste for the Extraction of Polyphenols and Polysaccharides of Commercial Importance. Her contribution to scientific literature is reflected through uh, numerous publications in various reputable journals, and her expertise is recognized nationally and internationally through various research that she has done and various review articles that she has been reviewing in protoplasma, plant cell, tissue and organ culture, clinical biotechnology, and Palestinian Journal of Nanotechnology. These are few of those prestigious research journals, but these are not only limited to all this. There are a number of more research reviews she is doing as of now. Moving to her accolades, she has bagged 3 research presentation awards in international conferences for her research in nanoscience and biotechnology. She has also won a silver medal for her research on the topic that she has done on, Bacillus sp isolated from fish waste and testing the application of extracted proteins in the cleaning of contact lenses. Now, as we know, as we have gone through her profile, research and teaching is her passion. And thus, I request our eminent speaker for today to please take over. And we are extremely excited towards hearing you, Dr. Shruti. So I'll pause here and I will hand it over to you. I'll stop sharing my screen so that you can pull up your slides as well. With this, please take it forward. Thank you so much.
Thank you, Dr. Gaurav, for such a in-depth introduction, I would say. I was very flattered. I hope you can hear me properly.
Yes, yes, Dr.
Yeah.
Shruti. Yeah.
Good morning to everyone. I'll be sharing my screen now. And just a minute, so that there is no hassle, I'll first share the screen. Let me know if you are able to see it properly. Yeah, it should come now. Is the screen getting shared?
Yeah, Dr. Shruti, we can see it.
Yeah. Like I'll put it on a full screen mode now. Like I told you all, and Sir has all rightfully introduced me and informed you that I'm going to present a topic called Biostatistical Insights, Elevating Food Technology Research. So now the first thing that will come to your mind is, oh my God, this is biostatistics or it is statistics. So what happens is we kind of feel a bit of inhibition when it comes to statistics. So most of us do. Since I come from a background where statistics was touched upon during my education in only one of the semesters, I would like to say that it's like developing a taste for it. It's like usually people are not that fond of apple at the first go. But what happens is if you eat apple regularly, you develop the taste for it and you start liking it. At least for me, that's what happens. So the statistics is something like that. And my journey with statistics started in ICT when I was doing my PhD. I got the golden opportunity to apply a lot of statistics, learn through my peers, my guide, and I had to put in some extra efforts from my end also to understand what it is all about. But once you understand the entire picture, you kind of understand why it is so important. So today's talk is going to basically enlighten you about different statistical techniques in food technology research. So I'm going to give you some examples and going to guide you in terms of how to go about applying statistical tools. So the do's and don'ts and everything will be covered today. So we all know what statistics is. Within that, we specifically call whenever we use biological samples or we use statistics in biology, we call it biostatistics. So when it comes to food research, if we are kind of finding antioxidant activity of some extract, like when we say fruit extract or when we say maybe a particular type of flower, edible flower, then what you're working on is a biological sample and you're trying to analyze some bioactivity in it. Hence, the statistics, though it remains the same, we term it as biostatistics. So, as you can see, look at this thing that I have written. I'm sure those who have just read it, it doesn't make any sense. Providing nutritional into the cornerstone and biostatistics food pattern is insights in through data research quality analysis. It doesn't make sense. That is why I purposely kept it like this, because this is what is meant by a raw data. The data that you collect through your experiments. It makes sense. Every word here has a meaning, but you have to analyze it, rearrange it, and present it in such a way that the entire community, the entire world can understand what you're trying to convey. So that is why there's a famous quote that statistics, or I would say biostatistics, is the grammar of research and science. So that's how it is. So if I just turn everything, analyze this, what this whole sentence is supposed to mean, and then present it in the rightful way, this entire sentence turns into biostatistics is the cornerstone of food research, providing insights into nutritional patterns and quality through data analysis. So it's not like your data is wrong or right. That comes later. But when you have a raw data, you cannot produce or present all the numbers to everyone. It's very difficult. If you work on about 500 samples, you can't write the values of all 500 and reflect it in a research paper to communicate what you have found out across the world. So that's difficult. So that's why it's necessary for you to describe the data in a concise manner and present it to the world. And that's where this statistics concept comes into picture. Another thing is, just by looking at numbers, you will never ever be able to conclude anything. We'll be coming to that. So before we move to that, I have here right now on this slide presented some examples, because when you all are from food science and technology, so when I give you examples related to food science and technology, it will become easier for you to grasp. Yes, statistics requires reading, no doubt about it, but today's session is going to enlighten you about the different directions that you can adapt for getting an answer to your raw data analysis. So whenever you want to do any kind of antioxidant assay comparisons between different samples, or you want to check the phenolic content, or you want to check the viscosity, or even the sensory analysis that you do. So when you have sensory analysis and when you have some data presented in front of you, we normally show it in a graphical format. But when you support it with a statistical tool and say that, okay, I have found a statistically significant association or difference, that is going to make a lot of difference, and it is going to increase the validity of your work, the weightage of your work, the reliability of your work. So moving on, I have written certain examples here. Suppose you want to evaluate the effect of increasing concentration of fruit extract on the acidity and sensory acceptance of a product. Now, in this case, what will we end up doing? We will use fruit extracts of different concentration do a particular assay to check the acidity of it, and also perform certain trials to see what is the sensory acceptance of the same. And then you just can't present what was the hedonic scale. I mean, how many people gave an excellent review, how many people gave a very bad review. That is not the way to present a data. Whenever you get such things, you are supposed to convert that into a particular type of variable, so that you can do statistical analysis and see whether at this particular concentration, the difference that you're seeing in terms of sensory acceptance, whether that difference is actually there is a meaning behind it, or is it just comparable to any other concentrations of fruit juices? Because people will respond to all the concentrations. You will be claiming that this particular concentration, the acceptability was the best, but that particular statement that you make needs to be supported by your statistical analysis. It is as simple as that. Any statement that you make in terms of your result and discussion, it has to be backed by a statistical tool which proves that there is that association or relation. So the second example I have given here is assessment of effect of food actives on certain biochemical markers like oxidative stress in the body. So nowadays there are nutraceuticals and functional foods coming up. You all are very well aware of it. So now when you say that this particular food item is good in acts as an anti-cancer agent or something of that sort, or it helps in preventing certain types of inflammation, you will have to do an assay. You will do that assay beautifully. You will standardize it. You will get the results. Now, the values that you get, you will have to compare those values. You will be trying the effect of that food active at different concentration and reporting one concentration as the best one. So that value that you get, you will have to show in terms of statistics why it is so good. So moving on, use of statistical tools in food research and development is important both in academia and industrial research. It is not like you need statistics only when you do industry-oriented research. Even when it is an academia-based research, maybe within a short span of time, it might not get extended or translated into a big industrial-based research. But nevertheless, it forms that particular cornerstone for someone else to work on it and scale it up. So what you do is going to be used by someone else to do something bigger. That's why it is your own responsibility to do honest work and do the right statistical analysis to back your data. So what will happen if you choose the wrong data? So choose the wrong test. So first thing is, I told you have to do statistics, but that doesn't imply that you can do any type of statistic. For example, you will first have to know what type of data you have. Based on the type of data, you will have to choose the right statistical tool. So I keep telling everyone that I know, it is also about the choice of the right statistical tool. Now for this, you will have to read certain basics rather than directly going to a research paper and seeing, okay, their work is similar to my work, so they have done this test, so I'll also do this test. That's not the way to go about. You should understand first thing whether what type of data you have got. So I'll explain that also, because what will happen if you choose the wrong test, you will get the wrong answer. So that is very important. This will lead to misinterpretation of your data. So there are many statistical tools out there that you can use. The free of cost is the R programming one. I personally use SPSS because I have a licensed version in my institute. So we use SPSS. There are other statistical softwares that are available. So you don't have to manually do any calculation. You only have to feed the right information and give it the right command saying that, okay, do this test. So for example, when you want to compare mean values, suppose you want to compare average weight of children in a clinical trial, suppose you are a part of it, and you want to compare the average weight of some group of children who were given a particular type of functional food or a nutraceutical. You want to compare that average weight of that group to another group who were not on this type of a nutraceutical or functional food. They were just having their normal diet, day-to-day diet. So after 6 months, what have you got? You're comparing the average weight, or it can be an average height also. So the point is that when you're comparing this average with this average, you want to show that, okay, the average weight gain was better in your particular test group to whom you administered that nutraceutical or functional food. And you are saying it is better than the nutritional profile of this kid, I mean, this group of kids who are on normal diet. So now the thing is, when you want to compare such average, you are supposed to go for a particular test. So the test that you normally go for is the independent two sample t-test or unpaired t-test. But many a times I hear many students who approach me say, I want to do ANOVA. I want to do ANOVA. So that's not how it is. You do ANOVA when the sample size is more than two groups. So there are certain rules which if you read any book or refer to any resources, you will get this data. Like when should you go for ANOVA? When should you go for t-test? So I'll be educating you guys about this. But remember, this is how it is. You cannot choose the statistic first and say, I want to do this statistic in my research. No. Whatever data you have, depending on that data, you are going to choose the statistical tool. So moving on, like I said, it is essential to understand your data set designations to carry out appropriate analysis. So poor quality data means if your sample size is very less. Suppose you do sensory analysis or maybe 5 individuals or 10 individuals. That is not enough. Maybe for maybe a dissertation, you can claim it to be a part of one small chapter in your research work. But if you're going to sell that technology or that way the processed food is made, and you are going to claim that its acceptability is good, its taste is good, its mouthfeel is good, aftertaste is good, we got good reviews. If you're going to claim that, you cannot work on 5 or 10 individuals and say, this is what I've got, so my product is good, so I'm not now going to translate it and we are going to patent it and all. I suggest the sample size has to be strong enough so that you can do statistical analysis to prove that there is a difference between your new processed food item and the existing processed food item. So now we move on to certain basics before we move on to the different types of statistical analysis that are out there. So before I move on to this, I just want you to remember the types of data. I have not kept them as examples or a separate slide. So first type of data, because you need to know this. So first thing is there is something known as qualitative data, and the next one is quantitative data. So when it comes to qualitative data, I'll give you an example of any feedback form. So in feedback form, when you are asked whether you're a male or a female or other or whatever it is, later on that information is presented in the form of a bar chart to indicate what was the gender distribution. So in this case, the data that is reflected, that is called qualitative data. Okay, so here this is nothing but nominal. It doesn't have any measurements. You're just claiming whether you are, for example, employed or unemployed, or marital status, married, unmarried, whatever it is. So whenever such description comes and it has no measurement or any kind of ranking involved, it is called nominal data. Why am I saying this? Because when you go ahead and when you open that chapter of doing statistics, first thing is you will see people will be writing in the books and all, there is categorical data, there is continuous data, there is ordinal data. Such terminologies will be used and you might get scared. So why am I doing this now? Because as we go ahead, I'm going to use these terms. So it's better that we all are on the same page. So one is that nominal, where it's all about the gender, male, female. You can think of it like that. The next one within qualitative comes ordinal, where the description is going to be in terms of some words. For example, you are given feedback form where you are told describe the session, and it will be good, very good, excellent. Then there will be poor, or fair, or poor. So here, though the description is in terms of qualitative data, but you are kind of ranking it, right? We know poor is the lowest ranked, fair is the second lowest, so on and so forth. And the highest would be excellent. So that is nothing but again qualitative data because it's not a measurement, right? But you are kind of ranking it. So this is called ordinal data. And moving on to the next one, which is going to be the quantitative data. When I count the number of people, when I say the number of people who said yes, who had lactose intolerance were like 150. So this type of data is called discrete data. It is a quantitative data. And the next one is going to be continuous, which is nothing but measurement, height, weight, so on and so forth. You need to know these terms because if this is clear to you, then whenever you read any kind of statistical book to understand what tests you're supposed to refer to, it becomes easy to grasp what the author is trying to convey. So now we move on to there are two types of statistics. One is going to be called descriptive statistics. The other one is called inferential statistics. So first I'm going to give you example food technology examples in descriptive statistics, and you'll understand how these statistics are very important because you read it in research papers, but you don't know sometimes what it is all about, and we get scared. So first, when it comes to descriptive statistics, it is nothing but description of your data. So when we say the vitamin D, average vitamin D level in this Indian subject group was found to be so much, or you say the average glucose level in this cohort of diabetic patients residing in western region of India was found to be about 180 mg per deciliter, the average. So what are you saying? You're just calculating the mean and expressing it. Why? Because the reader would want to know. No? If you directly go on to explain, or you'd say that, okay, vitamin D levels were compared between 3 different groups or something of that sort, and you directly jump onto the main inferential statistics, the reader would want to know what was the average age of the individuals of those 3 groups. They would want to know what was the average body mass index of the 3 groups, everything that is related to the vitamin D levels, right? So such type of information needs to be given. And you will see usually in research papers, the first paragraph is all about that in terms of result and discussion. So this statistic is called descriptive statistics. So vitamin D levels in a group of Indian subjects, the average vitamin D level, that's nothing but descriptive. You are going to calculate the mean and reflect it. That's it. So now this is another uh, uh, information that I have like copy pasted from this paper, which I have already cited here. It is from a renowned scientist from the Food Engineering and Technology Department of ICT and her team. So as you can see, mean will always be accompanied. Just 1 minute. Mean will always be accompanied with something known as standard deviation. So this particular research was about an extraction of some metabolites or actives from marigold flower. So Metagetes erecta was used, and coconut, and the shreds of it. And they have used waste, if I'm not mistaken. They have done supercritical carbon dioxide based extraction and of the actives. And then they have characterized and used it in a food formulation. This is what this particular food technology research is all about. So here, the paper essentially starts with first the description of what is the average yield found out at using different extraction parameters. So there are different extraction parameters. If you look at the table closely, you will understand pressure, temperature, time. There are different extraction parameters that have been used. Permutation combinations have been used. And the data basically reflects the mean value in terms of yield, and this very value that comes after plus or minus, that is nothing but your standard deviation. So this is very important. You need to reflect your data with either standard error of mean or standard deviation. I usually use standard deviation because look at the name. It says deviation. So it tells you how deviated the values are around a mean value. So this is a bit technical, but it's as simple as this. You can maybe try it out later. If I give you 5 set of values like 1, 2, 3, 4, 5, and tell you to calculate the average, you will get the average value, right? So, and at the same time, In another group, the value that I give them is 1, 2, 3, 4, and then 12. You will see the mean value, the average value shifts towards the higher side. That shifting has occurred because you have one extreme value, that 12 value. So that type of error, I would not say, but that type of deviation happens when we do research. Our 1 or 2 values deviate to either the lower side or higher side. And then what happens? Our average value shifts. So what happens? Suppose you have the same thing happening in vitamin D levels. I'll give you an experimental model. You'll have to pay a little bit attention to what I'm trying to say. Maybe it will be easier for you to understand. Again, the vitamin D concept we'll use. We have a control group and we have a test group. The control group individuals were on normal dietary regime. They were, and the test group were on normal dietary regime, and at the same time, they were given some vitamin D supplementation also. So after 6 months, you're trying to see what is the vitamin D level in the control group and the test group, right? So for this, you will calculate obviously the average vitamin D level of the control group, the average vitamin D level of the test group. Now the thing is, what happens is, suppose you're working on like 500 individuals or 1,000 or 2,000 individuals, depending what sample size you're supposed to use. In this case, what can happen is that your suppose you might get a vitamin D level here as a particular value, and the vitamin D level here looks extremely high. So which indicates that the vitamin D level has gone up, at least when you look at it. But like I said, just by looking at the two numbers, you cannot claim that the vitamin D levels have actually gone up. So suppose here it remains like 5 and here it has gone to about 45 or something. So what I'm trying to say is, you'll be like, oh yes, it is working. My vitamin D supplements are working. You will have to compare the means. You'll have to compare the sample size. You'll have to compare the standard deviations. What do we mean by standard deviation? Maybe here you have, out of the 500 individuals, maybe about 100 or 50 individuals had extremely high vitamin D values. The vitamin D values must have not increased that high in all the individuals. It increased in maybe 50 individuals. And because it increased drastically, when you calculate the mean, the average went completely, it shot up. So here in this case, what happens is, this number that you have got, the high, big number, that number is influenced by those extreme values. Understood? So that very thing that is happening, that needs to be assessed. So it's not just about the sample sizes that you have used in the two groups, but also about the individual values that are there. Average will tell you that, but standard deviation will tell you how deviated your mean is, how deviated your values are around the mean. So that's why this is very important. And whenever we do inferential statistics also, SD plays a very important role. So now we move on to the next thing. So like I told you, let it be an anti-diabetic drug or a vitamin D level enhancement pharmaceutical or nutraceutical drug, whatever it is. First thing, just by looking at numbers, you cannot claim that your particular drug or nutraceutical is efficacious. You will have to prove statistically, taking into account the deviation, the extreme values of each group, the mean value, also the sample size. All these things are need to be factored. So that is why we need to do inferential statistics. Remember, just by looking at numbers, you cannot claim that, okay, I found using anti-diabetic drug in the test group, the the sugar values went down by 40 mg per deciliter or something, and you claim that the difference is so much. Or test group, control group जो diabetic ही थे, and you did not give them any anti-diabetic drug. In that case, there was no decrease in or there was any insignificant decrease in the mean blood sugar level. And you cannot make this statement without backing it up using statistics. Okay. So before we move on to the inferential statistics, I just gave you a brief explanation of descriptive statistics. Just explained to you about two types of descriptive statistical tools, where one is standard deviation, the other one is going to be mean. In addition to that, what is the next thing that you need to remember? This is very important. Whether your data follows normal distribution or not. So I told you, first thing is, you have to first grasp what is your data, whether it is quantitative data or qualitative data. That is step number one. Second, you have to understand what you want to do with that data. How you want to compare it, whether you want to see whether with this value does the other value also increase, or you just want to compare the two groups and their average values. You have to understand the question first. You have to frame the question. Nobody else is going to help you in that. You know, when you're working, why you're doing that experiment and why you're using multiple samples. So that is one thing. Always remember what is the question you're asking, or remind yourself, this is the question I'm asking. So accordingly, I'm going to choose the statistics. The next thing that you need to understand is what type of data you have. And the third thing is whether the data is normally distributed or not. Because I need to explain you this, because many a times I see normal distribution is not accounted for, and directly t-test or ANOVA and all these highly efficient tests are done. They're efficient provided your data follows a particular distribution pattern. This is how what it is. So even if you this session may not help you per se in understanding all this at one go, but at least you can jot down the flow of it. And as you do go ahead with your food research, you will understand how to apply this. So this is the next thing is hypothesis testing. This is nothing but you will have two types of hypothesis for every kind of research that you do. First one is called null hypothesis, which means there is no difference observed. When I spoke about that vitamin D level between the control group and the test group, the null hypothesis would be there is no significant increase in the vitamin D levels in the test group as compared to the control group. And what would be the alternate hypothesis? There is a significant difference between the two. Okay. So this is like the foundation that I'm laying right now, because as we go ahead to inferential, you need to understand this, and then you will understand the application of it in food research. So the basic that thing that I want to really talk about is the p-value, because many a times I see the p-value is often misinterpreted. So there's a huge explanation behind it, but I'll just concise it and explain you, because whenever you read any research paper now or in the future, you are going to see that finally statistics is reflected in the form of what? P-value. And that p-value holds the most important, like highest level of regard when it comes to statistics. So if we say if the p-value is less than 0.05, that means the data, the difference is statistically significant. A very standard line which is always used. जो difference दिख रहा है आपको control और test में, if your p-value comes less than 0.05, it means that there is a statistical difference. So what now you need to understand is not why it is less than 0.05, because that would be another session altogether, but understand the definition of p-value. Because by hearting this line rather than doing that, what we need to understand is what p means. P means probability. Probability of what? Probability of you seeing some difference by chance. By chance means what? By luck. We say na, by luck. अरे ऐसे ही दिख गया हमें. So वो क्या था? तुम्हारा luck है. So would you want your research to have any kind of data difference that you're seeing to occur by luck? No. We would want it like, no, it is because there is some difference there. There is something experimentally happening, and that is why I'm seeing that difference. Suppose it's your processed food and you have enhanced its particular activity or its particular carotenoid content or phenolic content, and you want to show that your processed food that you have works better than another type of conventionally marketed processed food, or maybe a normal day-to-day food item. So you want to claim that. So in that case, would you want the statistical data to be by luck, the difference to be by luck, whatever you're trying to analyze? No. You would want that difference to be there, not by chance, but because there is something that you have done well, right? So this is how P is. P defines what? The probability of you seeing something by chance. So lesser the P value, it means lesser are the chances of me seeing something by luck. That is why if P is less than 0.01 or 0.001, people are very happy. We say it is highly statistically significant. So that is the meaning behind it. If you get a P value as 0.20, then in that case, the chances of you seeing the whole experimental difference uh, by luck is very high because it's 0.2. But if it is less than 0.05, it means that there is like, Baba, kuch to maine acha kiya hai, isliye wo farak dikh raha hai. So I hope you're understanding what P means. So this is the summation of what are the steps involved in hypothesis testing. You perform your experiment, collect the data, state what the null hypothesis would be, no difference, and alternate that would be, yes, there is a difference. Then select the right test statistic. That is the most important thing to do. The most brainstorming session that you have to do is going to be in selecting the right test statistic, okay, and calculation of the test statistic. Then the decision, based on the decision, you'll be writing the discussion of your paper. Okay. So now, like I told you, for after you understand what type of data you have to select whether the data is normally distributed or not. So this is something that is done statistically. I'll directly go to the main thing. So there are many tests that can be done. So there is this Shapiro-Wilk test that is there on SPSS and on these softwares. In addition to that, you have other also which are there, which you can do. So basically, what a data values, okay, should follow a normal distribution curve. So suppose you collect vitamin D levels, or maybe you check antioxidant activity in maybe the 20 cultivars of strawberries, and you're checking the antioxidant activity. Then in that case, the data that you have, it has to follow a bell-shaped curve. You don't have to plot that. You have to just see whether your data follows normal distribution or not. This bell-shaped curve is called normal distribution. So always remember, the takeaway from this is not to like by heart what I'm talking to you in terms of the analysis part, but rather understanding whether the data follows normal distribution or not is very crucial. Because based on that, you will be selecting which test you are supposed to perform. If your data follows normal distribution, then you go for t-test. If your data doesn't follow normal distribution, then you go for the non-parametric version of t-test. So if the data follows normal distribution, then we say that I will do parametric tests. There are many tests under parametric tests. I'll be using parametric tests. But if my data doesn't follow normal distribution, I cannot go for t-test. I'll have to either do some data transformation or something, or I'll have to directly go for what? Non-parametric tests. So this is all about the choice of the tests. You need to know this first. And for knowing this, you first have to analyze your data in terms of normal distribution. It is a mandatory thing that you have to do. So blindly, you cannot say, I want to do one-way ANOVA. You'll have to see whether your data follows normal distribution or not. If it doesn't, there is a non-parametric version of one-way ANOVA, which is called Kruskal-Wallis test. Then you go for that. And all these tests are there in your softwares. So that is what I wanted to highlight. This is the most crucial slide, I would say, because let it be medical biotechnology research, population genetics, let it be even food technology, food science research, this remains the same. Okay, the way to use this depends on what you're doing. For example, I've written the uses here. So if you're testing the difference between two independent groups, control and test group, like I told you right now. If it is control and test, that means here there are maybe 100 individuals and here there are other 100 individuals, right? You're comparing the average vitamin D levels of the control with average vitamin D levels of the test. So there are two independent sample groups. So in that case, you would be going for what? Unpaired t-test, two sample t-test. So when there are more than two groups, then you go for what? ANOVA. and so on and so forth. This very thing that I've written here, this is the thing that I will be discussing in detail now in inferential statistics in terms of food research applications. So let us now summarize the whole thing, because these are your basics. You need to know that you need to do this before applying some actual inferential statistic. That is why I wanted to talk about this first. Otherwise, directly jumping on and showing you the relevance of statistics and importance of that in terms of good published research articles and its application will not make sense. You have to first understand the essence of the whole statistics, how to start, what is the foundation, what all things I need to take care of. It's like you cooking something in kitchen, so you can't directly start with just taking an onion and just throwing it in tail. What do? We collect all the ingredients, we see whether they are ready, they're proper, they're there in proper amounts or not, and then we start with our cooking session. I mean, it's when it think about baking a cake, or maybe think about cooking a vegetable or anything of that sort. So you cannot randomly just go a barge in and just add something. What do you do first? You first methodically see whether these things are there or not, right? That's nothing but your data. Whether all the data is available or not. You will check that out. Then you will start with the initial preparation, which is nothing but cutting of your onions and tomatoes and coriander leaves or whatever it is. You'll wash them, you'll cut them. You will not directly take like half an onion and just throw it inside, right? So I'm just creating this analogy to tell you in terms of statistics what we do. We collect the data, we see whether we have the data or not. Then what do we do? The initial preparation. We are going to see whether the data follows normal distribution or not. Then we'll see what type of tests I can do. And then the main action part where you feed everything and you give that command to SPSS or SAS to do a particular statistics is nothing but your cooking. I hope you have understood the analogy. So understanding from this perspective will give you a better idea, because when you do it in a very methodical way, the sky is the limit when it comes to understanding statistics. You will be hungry to learn more. Trust me, I've been there. So initially I was extremely, how can I say, inhibited in terms of doing statistics. I was very scared. I'm telling you, in 2009, I got enrolled in 2008. In 2009, the chi-square test was the first test that I did, and I was very scared, very, very scared. But then slowly, like I told you, I started eating. It's like eating an apple. You develop the taste for it. Then I developed the taste for statistics as I started learning more. And once you get a software and you can feed the data, you can just check what is going on. The sky is the limit. I mean, if you're not getting the right answer, then you understand what is going wrong. Maybe you have to go to back to square one, do some certain experiments or repeat it all over again. It's perfectly okay, because as you're doing your experiments, you can simultaneously check what is going on. So this is a summation of whatever we have discussed so far. So here we have parametric tests and non-parametric tests when to do. So suppose you have an experimental data. If the samples are only like 2 samples, doesn't mean those sample hai, it means 2 sample groups. Okay. You will check first for what? Normality. Let it be 2 groups or maybe 3 groups. You are going to check for normality. Now, what we mean by 2 groups and 3 groups in terms of food research, we are coming to that. The next thing that you will check is check for equal variance or not. So that is this particular test. The next is once you check for these two, this is the initial preparation. Then you will decide which statistical tool you are supposed to perform. So like I said, if your data follows, the 2 group data follows what you say normal distribution, you will go for your t-test. And if it doesn't follow normal distribution, then you will go for its non-parametric version. Similarly, when you have more than 3 sample groups, you cannot go for t-test or manually. Then in this case, you will apply again normality to understand parametric or non-parametric. If it follows parametric, then you will apply ANOVA. If it doesn't follow normal distribution, you'll go for the non-parametric version of whatever test you initially wanted to perform. Normally, we get attracted to ANOVA and t-test and all, but nevertheless, you don't have the liberty to do that. You'll have to check for the normality and the variances. Then you'll have to do the data transformation or go for the non-parametric version accordingly, and then take your call. So now coming to the main action part, which is going to be statistical techniques, okay, how to perform them and in what ways they are performed in food research. So first is I have divided this into two sections: exploring relationship among variables and exploring difference between groups. So it sounds a bit confusing. Don't worry. As we come across certain examples, you will understand that. Yeah, I'll just have a sip of water, if you don't mind. Just a minute. Yeah, I'm back. So here, specifically, let's start with the discussion. So like I said, first thing, when you want to compare mean values. So when you want to compare mean values, what are you going to do first? You are going to decide how many groups you have. Okay. You will decide how many, suppose you have two extracts that means two groups. Okay, two fruit extract or two edible flower extract, it means you have two groups. Then you go for a t-test. If you have more than two groups, then you go for ANOVA. Now within ANOVA as well as t-test, you have what you say, many versions within that. You have paired t-test, unpaired t-test. You also have, in terms of ANOVA, one-way ANOVA, two-way ANOVA, repeated measures ANOVA. So the thing is, I'll try my level best to explain when what is used. No doubt about it. But let me see how it goes, and then we'll interact and just come to a consensus regarding. Maybe I just read side by side some messages written in chat. So definitely I'll help you guys out in terms of which books are good for understanding the basics of statistics and all. So let me just move on to the next slide. So we are now going to start one by one with different statistical tools. And I'm also going to write the non-parametric version of it. I hope you have understood what parametric and non-parametric means. So first, exploring relationship among variables. Variables means what? Measurement of temperature, measurement of vitamin C content, or any measurement that you're doing in your study. I'm going to put it in a very easy way for you to understand. Because if I try to complicate it by using a lot of statistical terminologies, not everybody will be able to understand. So in order to maintain the parity, I am just toning it down to this kind of language where I'm saying measurements. Okay, so you measure DPPH through DPPH assay you calculate the percentage scavenging activity. You're calculating something. You're finding a measurement, right, for that concentration of your extract, or you're simply measuring the weight, or you're measuring the vitamin C content, or you're measuring vitamin D levels, or measuring glucose levels. What are we talking about? Variables. Your glucose level is a variable. Vitamin D level is a variable. Vitamin C content is a variable. An antioxidant activity is your variable. So now suppose your objective, you are a researcher who is now trying to work on two different varieties of strawberries, and you want to analyze what is the vitamin C content in these two cultivars of strawberries. That's one of the objectives, not the only objective of your research work. Now, when you want to do this, what can you go for? What will be the situation? You'll have two groups, one of one strawberry cultivar, the other one of another strawberry cultivar, right? Now, what are you going to do? You will have average vitamin C content of, are you going to work on one strawberry only? Suppose it's from Mahabaleshwar, and one is from Mahabaleshwar, and the other one is from another region where you have tried to mimic the conditions and you're trying to grow a strawberry. Okay. So now the thing is, you are trying to see how well your vitamin C in this new condition in this new area where you're growing strawberry, in terms of vitamin C content, is it the same as the strawberries available in Mahabaleshwar? So here, what will you do? Are you going to just pluck one strawberry or get an extract from it and calculate the vitamin C content? One strawberry from the region X, wherever you have transplanted some crops of strawberries, plants of strawberries, and you are growing it, and the other one is from Mahabaleshwar. Are you just going to pluck one strawberry from Mahabaleshwar? No, you will take multiple samples from one particular area only, and you will calculate the average vitamin C content of strawberry from Mahabaleshwar, right? Similarly, you will calculate average vitamin C content of the strawberries grown in this new area. And what would do now? Are you just going to see the vitamin C values and comment on it? No. You'll have to compare the vitamin C values. Now, this is where you'll have to perform a statistical test. Now, when you have two independent groups, like I told you, two strawberries from two different regions, and you want to compare one variable, which is vitamin C content, then you go for what? You go for independent sample t-test, also known as unpaired t-test. So there are different terms used, two sample t-test, student t-test. Student t-test is a general term. I would say people end up using independent sample t-test or unpaired t-test as terminologies for this type of a statistical tool. So please remember that. So second example that I've given is where you want to compare enzyme production by two different bacterial strains. Enzyme production in the sense you want to extract a protease from two different bacterial strains. One might be a naturally occurring version of E. coli. I'm just giving you a general example, okay? E. coli, or let's take E. coli because that's the first organism bacteria that came to my mind. And the second one is a genetically modified E. coli. Okay, so you are working on that to enhance the production of protease in that particular bacteria, that particular strain of E. coli. Now, in this case, what are you going to do? You will obviously, since protease is your aim to increase the production of protease, or maybe you're not genetically modifying it also. You are optimizing the growth conditions and in such a way that you, through RSM and all, you are going to do that, and you are going to end up getting a protease. And this protease that you get, you are obviously going to carry out some assay, isn't it? You will carry out some assay to determine what is the protease activity. Now, in this case, what is the variable? The protease activity assay. So you will have again two groups. One is this particular naturally occurring bacterial strain. The other one is the new isolate that you have, which you have optimized in terms of growth conditions, or it is the genetically engineered one, whatever you want to label it. When you're comparing the average protease activity of these two different bacterial strains, then what are you doing in this case? When you are doing independent sample t-test. Remember that. Now, like I said, I have directly told you the name of the parametric test. But remember, before choosing independent sample t-test, first, whenever you get that average vitamin C content, you have the raw data. Now, you will have multiple strawberries that you will use from a particular region, and that multiple strawberries used from Mahabaleshwar. You will have that raw data with you. The data that you have, you are going to basically determine whether this data follows normal distribution or not. If it follows normal distribution and you wish to compare vitamin C content between these two groups, you go for what? Unpaired t-test. If the data doesn't follow normal distribution, don't worry. You can go for the non-parametric version of unpaired t-test, which is called Mann-Whitney test, Mann-Whitney U test. So I hope you have understood how it goes about. So this is about unpaired t-test or independent two-group t-test. Remember, here the prerequisite is you should have two separate groups, okay, the categories. So here the two groups I have mentioned what you need. You need two groups, two cultivars of strawberry or two bacterial strains. In case of humans, a control group and a test group. In case of what are you comparing? You're comparing vitamin C content or you're comparing antioxidant activity, or you're comparing the protease activity, that becomes what your comparison factor. So that's your variable. So now let's move on to the next way, another kind of statistical tool. Here, you suppose the research objective is to study whether the antioxidant rich dietary intervention in subjects, okay, whether it is helping in mitigating means addressing or lessening the effects of some metabolic disorder. Suppose let's take into account diabetes or something of that sort. So, so it's a metabolic disorder. So here in this case, what is going to happen when you want to do this? So you will have a same subject group, and that subject group that you have, that subject group, you will check what is the so since I said diabetes, you will check the glucose levels in these individuals, right? And then you will give them that antioxidant rich dietary intervention for maybe 1 or 2 months or maybe a month. And after that, you will check what is the glucose level. So here, what is happening? What do you want to do? What is the question that you are asking? Whether my antioxidant rich formulation, whether it helps in mitigating that particular problem. So here you will have the same group. You will have before treatment, what was the mean blood glucose level in those people, and after giving the antioxidant rich dietary intervention, what is the blood glucose level in those individuals. So it's as simple as that. So here, when you have the same set of individuals, but you have two mean values at two different timelines that you want to compare, in those individuals only, you go for what? PET test, provided that data that you have, those the average, not average, the glucose values that you have, maybe of 500 individuals, that data follows what? Normal distribution. If it doesn't, then you go for what? Wilcoxon signed rank test. Now we move on to the next part, which is, you all must have heard, ANOVA. So there are different types of ANOVA. I'm directly going to come to business. ANOVA is used when you want to compare mean values again. But the comparison now is not between two groups. It is between more than two groups. So I've given one example here. You will be able to associate with that, since you all are from food science and food technology departments. So rheological behavior of honey under different temperature conditions. Suppose you're checking the viscosity of honey. Now you will measure the viscosity at 25 degrees Celsius, 30 degrees Celsius, 35, 40, 45, and 50. Again, let me remind you, you're not going to focus on only one sample of honey at 25 degrees Celsius. You're going to focus on maybe 4 or 5 honey samples, maybe 10, 12, 30, 40 honey samples, and you will check the viscosity of all these samples at 25 degrees Celsius. Then similarly, you'll perform the same experiment using 30 to 50 degrees in that gradation, whatever has been mentioned. So you will have average viscosity values for each temperature. 25 डिग्री सेल्सियस पे एक एवरेज विस्कोसिटी वैल्यू रहेगा, 30 डिग्री सेल्सियस पे एक एवरेज विस्कोसिटी वैल्यू रहेगा हनी का. सो अभी क्या हो गया, यू हैव हाउ मेनी ग्रुप्स हियर? यू हैव 1 2 3 4 5 6 ग्रुप्स. In this case, you cannot go for t-test. You will have to go for something called as one-way ANOVA. Now you'll be like, why can't I go for that? It's another session, but all I can say is the error gets compounded. So that's what it is. There's a statistical explanation behind that, but explaining that would make this, we would be digressing from the main topic. So the only thing is, for now, remember, when you have more than 2 independent groups, you are going to go for what? One-way ANOVA. When you're comparing mean, remember that. The other example that I've given, I think the same examples are there in that table also, the tabulated form. Yeah, here. So here, suppose your research objective, the first one I've already discussed, which is rheological behavior. The second research objective is you have 5 different varieties of apples from 5 different regions of India. Now, what your aim is to analyze their average catechin content. So you have these 5 varieties of apple. For each variety, you will have an average catechin value. catechin content along with a standard deviation. So the apples here, the 5 apples are your independent variable. They are the groups, 5 groups. In this case, they are apple 1 from one region 1, apple from region 2, apple from region 3, from region 4 and region 5. These are what? These are going to be your groups. And the catechin content that you have found out by doing your research, by doing your assay, that catechin content is the measurement that you're comparing. You want to see whether the average catechin content of in these 5 apples, are they comparable, or is there any statistically significant difference between them? In this case, you'll have to go for one-way ANOVA, provided your data follows what? Normal distribution. If it doesn't, then go for the non-parametric version of it, which is called Kruskal-Wallis test. So I hope you have understood one-way ANOVA, like when it is performed, unpaired t-test when it is performed, and paired t-test when it is performed. So and like I told you, I have given some food technology-based research examples here so that you will be able to associate when you can use these tests and how you can use this test. And like I said, performing these tests in terms of how to, feed the data, you just, I'll give you an example of SPSS. Just, it looks like kind of like Microsoft Excel. You have the data imported into that. And then you have, in particular drop-down boxes, you can just select what test you want to do. And then within that, you just select the right commands and just tell it, give me the output. The output that comes, you have to analyze. In that case, for now, remember, if you want if there is difference, your p-value will come less than 0.05. It's like that. Now moving on to the next type of ANOVA, which is going to be two-way ANOVA. This is again, it's going to get a bit complicated. I completely understand. But remember at least in what type of research it is used. So here I have taken this from this very interesting research paper, which I have cited below. It's on antioxidant capacity, ascorbic acid, total phenols, and carotenoids. Just a minute. Carotenoid changes during harvest and after storage of this particular kiwi fruit. It was published in Food Chemistry. So here they have performed a two-way ANOVA. So two-way ANOVA basically is done when you have, how can I say, two categories involved, meaning I would like to frame it in a better way. Maybe I'll be able to. So in this case, what would happen is you would have what before and after kind of a assumption also. So this is a bit complicated, so I don't want to go into that. If you want me to explain it, maybe in the Q&A I can. Just to summate it, they have the ascorbic acid content in kiwi fruit. That kiwi fruit was harvested at two different times. It's written in the figure legend also. And it was stored for like 2 and 6 months at 0 degrees Celsius from what I've understood. So what are they trying to see? They are trying to see what is the vitamin C level, meaning ascorbic acid level, at the time of harvest and at the second month and the second month plus 7th day. So they're trying to see, it is what is a takeaway from this? You have the same kiwi fruit samples. The kiwi fruit samples at the time of harvest, what is happening to it? At the end of 2 period of cold storages, what is happening to it? So they will be taking some samples after 2 months and 6 months, and they are going to compare what the ascorbic acid content. So what they want to see is whether the storage time is influencing the vitamin C content or not. So again, let me repeat here. It's all about before and after I told you. So you end up having initially a particular sample of your kiwi fruits at the time of harvest. Now you're going to store that kiwi. So you will calculate the vitamin C content by taking some samples out of it. You will then store the remaining for 2 months and 6 months. So here the groups, if you say there are 3 groups: harvest, 2 months and 6 months, right? So here, what are you trying to do? You're trying to compare the average ascorbic acid content at the harvesting time, the second month and the third month, 6th month. So you're trying to compare vitamin C levels, not between 2 groups, but 3 groups. But what has happened here? understand that the sample is the same. आप वही कीवी फ्रूट का जो बंच है उसमें वो आप थोड़ा यू आर टेकिंग आउट ड्यूरिंग हार्वेस्टिंग टाइम, calculating the ascorbic acid. After 2 months, you're taking out some samples, calculating the ascorbic acid. After 6 months, you're removing and calculating the ascorbic acid. After storage at 0 degrees Celsius, whatever your parameter is. So here the group remains the same. This is like if you have unpaired t-test for 2 independent groups, you have one-way ANOVA for more than 2 independent groups, right? Paired t-test was done when you are comparing before and after. Like I give you the vitamin D supplement example, before treatment and after treatment. But when you have more than 2 after treatment regimes, then what will you go for? Two-way ANOVA. So it's like paired t-test ka higher version. So now moving on, there is another type which is called factorial ANOVA also. I just wanted to make you aware that there are different types of ANOVA. One-way ANOVA is not the only one out there. So depending on what you're comparing, you have to select that in that way the respective ANOVA, or its non-parametric version. This is what I want to highlight here. So factorial ANOVA is something that is widely used in sensory analysis. Now, the next one is going to be repeated measures ANOVA also. This is kind of similar to factorial, well, you can say that. But this, I made a mistake here. Two-way ANOVA and factorial are similar. The one example that I gave you about before and after, there are 3 parameters, that is repeated measures ANOVA. I stand corrected here. So now moving on to the next one, which is going to be correlation. Now, when it comes to correlation, what happens is you end up having a relationship between 2 variables. You're not trying to analyze whether the difference in the mean is making, is there a difference between the mean values? Not like that. You're trying to correlate, as simple as this, you're trying to correlate whether the weight increases with increase in height. We say now, as the age advances, the weight increases. As age advances, the bone mineral density of a particular age group increases. So यहाँ पे हम क्या करते हैं? We are doing correlation. Understood? So correlation is done when you want to establish relationship between measurements. The example that I've given here is correlation between phenolic content and antioxidant content of grape cane extracts. So suppose you have a particular type of grape cane. Take sugar cane if you want to. So in this case, what are you trying to see? Whether if the phenolic content is more, whether there is increase in antioxidant activity. Now you'll be like, how to perform this experiment? You will collect sugar canes from different areas, from different regions where of Maharashtra, maybe from where it is produced. And then you will determine the phenolic content in that sugar cane. You will determine the antioxidant activity also in that sugar cane. And what are you trying to see? Whether depending on the phenolic content or number of phenolic groups, अगर वो ज्यादा हुआ तो antioxidant भी ज्यादा रहता है क्या? This is what you're trying to find out. There's a lot of research going on in this line because we seek antioxidant activity, but people want to now pinpoint exactly what is contributing to that antioxidant activity. So polyphenols are supposed to be very famous for enhancing antioxidant activity. So if polyphenols means what? You have multiple phenolic compounds in it. So phenolic group ज़्यादा रहेगा तो फिर whether you are having more antioxidant activity. So if you're trying to do this type of analysis, you will go for Pearson's correlation coefficient. So remember that. Here you will have 2 continuous variables, meaning measurements, which is phenolic content. So you may be having about 100 sugarcane samples, and you calculated the phenolic content of 100 sugarcane samples and the antioxidant activity of those 100 sugarcane samples. Now here, what are you comparing? You're not comparing mean phenolic content with mean antioxidant activity. That doesn't make sense because these are 2 different variables. Remember, before this, whatever examples I was giving you, especially in unpaired t-test and all, I was telling you to compare vitamin D levels in control group with vitamin D levels in your test group. So that variable was the same. But when you are comparing 2 different variables, and you're trying to establish a relationship between the two, like I said, height and weight, temperature and pressure, or phenolic content and antioxidant content, activity, then what are you going for? You are going for correlation. You're correlating these two independent variables. एक वेरिएबल का वैल्यू बढ़ा तो दूसरे वेरिएबल का वैल्यू भी बढ़ रहा है क्या? This is what you're trying to establish. So there are different types of correlation. This is for your understanding. So you have no correlation, positive correlation, negative. So what will happen, guys, when you do your SPSS or SAS, you are going to get an output. Now I told you about p-value, right? So p-value can be applied to whatever I discussed till now. But when it comes to correlation, our focal point is this value, the R value. What is that R value? Is it 0? Zero will never come. That is an imaginary, a fictitious number in this regard. You can get it as 0.02 or something, or minus 0.02. So you will get a value like maybe 0.9, 0.5, 0.82, so on and so forth. The value of R will always lie between +1 and -1. Always. By default, it is like that because the formula to calculate correlation is in that way. The value will always land between +1 and -1. So here, depending on the strength of the number, the magnitude of the number, you will determine whether the correlation between the two variables is strong or weak. If the value comes as 0.9, which is very close to 1, and then you will say that the correlation is strong. Suppose you get a relation between a number of phenolic groups, phenolic content, and antioxidant activity of those 100 sugarcane samples. You did the correlation and you got an output of maybe 0.6, or I would say 0.9. Let's keep it as 0.9. Then you would say there is strong correlation. But the direction of the value also matters, meaning whether the sign is positive or negative. If the sign comes as +0.9, then that indicates that there is strong positive correlation. It simply means if the phenolic content is more, the antioxidant activity would be more. I hope this is clear. And when it comes to a negative, if the value comes negative, and if it is -0.9, then strong is the word you will use because the value is 0.9. It is close to -1, -0.9. But in this case, the direction is negative, which means inversely proportional. If one value is less, then the other value would be more. Okay, so this is what we mean by that. And then comes a very strong tool called regression. Now, when it comes to regression, this is used as a predictive model, meaning you can predict something knowing something. Okay, so I'll directly go to the main thing. Just a minute. Yeah. So example here I have given is, suppose you want to identify the relation between antioxidant property and total carotenoid content and phenolic content. You're not just trying to establish a relation between antioxidant property and one variable, but antioxidant property is itself one variable, and carotenoid content and phenolic content. You want to establish a relation between all this. So you cannot do t-test and all and just compare the mean values and state something here. You have to go for something bigger. The bigger thing that you can go for is regression, multiple regression. There's linear regression when you're comparing maybe only antioxidant property with total phenolic content. You can go for correlation also. You can do both. That is also okay. Let me tell you that. You can do correlation, give the Pearson's correlation coefficient value, because that will establish what type of relation is there between them. And then regression will tell you what is the strength of that relation. I hope you are understanding this. So normally we say correlation and regression. So this is how it is. So here, when there are only the two variables which you are trying to establish in terms of relation, you are going to do what? Linear regression. But when there are more than two variables, then you will go for what? Multiple regression. But now I'll explain it in a simpler way. So what do you need in this? You need one dependent variable and certain independent variables. Now look at the logic. Depending on my carotenoid content and phenolic content, Accordingly, I will have an antioxidant property, isn't it? Look at the statement. एक plant के अंदर या एक food produce के अंदर, how much is the phenolic content and carotenoid content is going to basically define what is going to be the antioxidant property. This is my postulation. I'm postulating this. So I have to show that there is a relation between all of them. So for this, I'll be doing what? Regression. Here, what is dependent? Your antioxidant is dependent on your carotenoid and phenolic content. I hope you're getting it. Your antioxidant is the dependent variable, which is depending on what? According to you, it is dependent on your carotenoid content and phenolic content. You'll be like, how will we come to know? Through experiments, guys. Through your literature review, you will be designing your experiment, you'll be stating an objective. So maybe this is your objective. You want to identify whether there's a relationship between antioxidant property and total carotenoid and phenolic content. If you want to do that, then you have to go for what? Multiple regression, or only if you want to establish between phenolic content, one independent variable and one dependent variable, then you go for linear regression. So this is the paper that you can refer to. This objective I have got basically noted it down from this paper published in 2023. It is on rapid screening of microalgae as potential sources of natural antioxidants. So this paper will be handy. It's very nicely written. So whatever I just now explained is just written here all over again. So I'll not repeat it. Then the final one is going to be exploring the relationship between among variables again. We discuss linear and multiple, but it's not necessary that every time my variables are going to be measurements. I can simply have qualitative variable, okay, maybe a nominal one. So let me just state it for you. Now, this is something that I worked in ICT. It was my PhD topic, one of the aspects of it. So this basically talks about understanding what is the risk factor of developing lactose intolerance. So what are the risk factors involved? So for this, I had to do a lot of population genetic studies, and it was on nutrigenomics and nutrigenetics. So here in this case, what is the dependent variable? What is the output? Lactose intolerance. I'm trying to establish in my case through initial analysis, what I found out was like age and gender and the genotype of the individual is playing a role in lactose intolerance. That's what my hypothesis was. Okay, my alternate hypothesis that there is an association between them. My null was there is no association between them. So I had my data in front of me. I had SPSS software with me. So all I did was just do this logistic regression analysis. This is another type of regression. When do you do this? When your dependent variable is not a measurement. See, look at what the output is. Whether a person has lactose intolerance or doesn't have lactose intolerance. So this is not what you would call a quantitative data, right? I mean, it is quantitative, but it is going to be discrete, like number of yeses and number of no's, right? So here, this is categorical dependent variable. So this is the kind of output my dependent variable has. I mean, it is not a measurement like you had antioxidant property. You will be doing DPPH assay or ABTS assay. You'll be getting some value from it, the percentage scavenging activity, some calculation will be doing. Here, there is no calculation. The answer was straight away yes for lactose intolerance and no for lactose intolerance. So here in this case, what do we go for if I want to establish whether age, which is another type of what I would say data like depending on less than 50 and greater than 50 years of age. That is the way the category was made. And then there is gender, which is another categorical variable, male, female in this case. That is what I had considered. This was in 2010. Then the third one is genotype. So in this case also, there were three genotypes: homozygous, wild type, heterozygous, and homozygous mutant. So I wanted to establish a relationship between these categorical variables, not measurements. So that's why I had to go for what? Logistic regression analysis. So like I told you, during my PhD, I got to learn this. And I only knew regression, But what I found out was that what I am trying to do doesn't involve any kind of measurement. Then I got scared. How am I going to predict whether if a person is greater than 40 and if a person is female and has this genotype, what are the odds of this person developing lactose intolerance? I wanted to do this. That's where I started reading papers, not related to lactose intolerance, but in general. So I stumbled upon this. Then I understood different types of regression analysis. I read about them and I came to know, I knew for sure I need to do logistic regression analysis. Had I done something else, because we will always have a number of lactose intolerance and all that, and I end up doing multiple lactose intolerance, then in that case, it indicates something that is absolutely wrong. So this is one important thing that I want you to remember. The second last, just a minute. Yeah. The second last thing that we need to remember in terms of analysis is going to be chi-square test. So in case of chi-square test, what do we see? When do we do this? When you're trying to associate something. Now I said establishing a relation, and now I'm saying association. It'll be like, मैम दोनों साइड से दोनों चीजें बोल रहे हैं. So ये दोनों साइड से दोनों चीजें kind of नहीं है. I'll explain what the whole thing is all about. So when I say association, chi-square test, the data is going to be discrete. Discrete means quantitative data, but how many people or how many, what was a shoe size or something of that sort. So let me just put it in this way for you to understand. So here, as you can see, we are exploring the relationship among variables. You have heard about correlation. Now you will hear about regression. So when you hear about regression, you were establishing a quantitative variable. Just a minute, I'll just erase this. Something has come. Yeah, it was distracting me. So yeah. So when you are trying to establish a relationship, there are different ways and you can do that. First is going to be correlation, where you have two groups, where you have measurements in them. Okay, antioxidant activity measurement and phenolic content measurement, correlation. When you want to establish relationships in further between them, you go for a higher version, which is going to be regression. The third one is going to be chi-square. But in this case, what happens is you have categorical variables. Matlab kya? You won't use measurements here. You will use number of phenolic groups. Can you see this? So if you want to determine whether antioxidant activity of flavonoid is influenced by the number of phenolic groups. So here it is not phenolic content. It is number of phenolic groups. So here it will be a count. Then you go for what? Chi-square test. It is as simple as that. Another example I can give you is if you're trying to associate lactose intolerance with milk intake, frequency of milk intake. Then in this case, what are you going for? You will be going for chi-square test. I'm trying to associate whether if the milk intake is, or maybe this a homozygous mutant individual, whether that has an association with lactose intolerance or not. So here, we are just going to count how many individuals were a homozygous mutant. You're not going to do any kind of measurement here per se. Measurement, when I say measurement, I'm talking about any kind of assay, right? And here, you're going to associate that with the number of people who are lactose intolerant and who are not lactose intolerant. So the answer is more like yes or no, or it is about how many phenolic groups were there. So how many, when we say that, it's a type of quantitative data, but it is a discrete data. It is a categorical data. Okay. So this is about chi-square. I have widely used this test in my study for like during my PhD. So it's very, very interesting. You should really think about this, even in terms of food science research. I don't see chi-square, at least the papers that I have visited. I have not seen a chi-square test for being done that often. But it is a very, very strong tool. It is, there's no non-parametric version of it because it itself is considered to be non-parametric. So this is it. Now, just a minute. Some of this not going to the next slide. Yeah. So we move on. We are done with all the inferential statistics that I wanted to explain to you in terms of, what can I say, food research to inspire you. I have been revolving around only antioxidants and phenolics, because what I wanted to show you, that there is no, you cannot say that well, this is the only test I can do. Depending on how your data is, you're going to choose the test. That's why I kept the analysis part that has been done the same, meaning phenolic content or antioxidant. So as you could see, the same thing could be compared in different ways depending on how you have your data with you. That's what it is. So now, finally, this is just a summary of something that you should do, which is graphical representation of data. This is for your understanding. I don't think I have that much time to cover this aspect of it. But nevertheless, if you have ungrouped data or group data, there are different types of graphical patterns that you can follow. So let me just go to the action bar. Ungrouped data means straight away measurements. Like, how can I say? If you say age, so age will be 18 years, 30 years, 40 years, 50 years, depending from person to person, it will vary. That is ungrouped data. But the same data, if I reflect it in this format, age 18 to 30 years, and I just write how many people were between 18 to 30 years, that is called group data. I'm grouping the data. So that is what we mean by ungrouped data and group data. So there are different versions of graphical representation of data. I just wanted to show you this one. I will just discuss this and then maybe we'll summarize the whole thing and call it a day in terms of my talk. So as you can see, what does this do? This is huge amount of research which has been done. If you look at it, there are different fruits and vegetable items which have been studied, and the total phenolic content in all these food items, fruits and vegetables, have been measured. Now, the paper that I have cited below, this paper is on storage of fruits and vegetables in refrigerator, and they're claiming that it increases their phenolic acid but decreases their total phenolic, anthocyanin, and vitamin C with subsequent loss of antioxidant property. So this type of research they have done. So what they have done is, in this case, look at it. They have used so many food items, measured the total phenolic content before storing it in the fridge, and then again measured their phenolic content 15 days later after storing it in the fridge at 4 degrees Celsius. And now the thing is, they will do their analysis. They have done elaborate analysis in their paper. But please understand that as readers, when we read paragraphs, yes, we will understand. But when we see the same information represented graphically in this way, in a concise, summarized way, it creates a lot of difference, because you come to know exactly what the author wants to talk about. Here it is loud and clear what is happening, like from before and after. Look at the bar size that is there. So that's what there is. Like, tabulated formats are also important, but graphical representation of data can concise and summarize your entire work into one particular picture and give the reader the idea, the gist of your paper. So remember, this is also equally important. You can make this on Microsoft Excel also, so there's nothing to worry about. The next one is a simple one, which is pie diagram. This is something that is used for mentioning what all distributions you're observing in your work. So this is, you can do the same using bar chart also. It's not necessary to draw pie diagram, just making you aware that something like this is also there. Then the next one is histogram. So like I had mentioned earlier, histogram is going to be used for group data. So please remember that. So just let me go there to show you. So when it's group data, you can go plot a histogram. So because the data is in a continuous format, like age group 18 to 30, then from 31 to maybe 40, then 41 to 50, it's in continuation. So the bars need to be close to each other. That's the logic behind it. Then there is another type of graph that you can show, which is called frequency polygon. Here, what happens is, the trend that is there, you can compare different trends. So your X, Y, and Z diseases are being compared. The number of cases in terms of X, Y, and Z diseases is being compared across a particular set of months in 2012. And what can you see here? Like just by looking at it, you come to know the trend of the disease. The green one is the Z disease, the red one is the X1, and the blue graph is the Y disease. You come to know that in the 7th month, the Y disease Y was the prevalence was the highest. So the frequency polygon kind of gives you that liberty, the comparison part. The next one, this I've taken from this particular paper, which I've cited. I liked it because it's kind of summarizes. This is like another version. It's called frequency curve. Here, what happens is the points that are joined, they are not joined directly to form in the form of lines, but rather a curve is drawn around it. So frequency polygon is one. The other one is frequency curve. Curve kind of shows you a trend, growth curve analysis of bacteria and all. So the trend line that you see in the curve gives you a better idea. So this work was on production of vanillin from ferulic acid using recombinant strains of E. coli. So here what they're trying to see is, look at the x-axis with the increase in time. With the increase in time, what is happening to the vanillin and ferulic acid content? So the vanillin content, I think from what I have understood, I've not read the paper in detail, the vanillin content is going up and the ferulic acid content is going down, indicating that ferulic acid is being utilized for forming what? Vanillin. So this is another study that I came across. So to summarize the whole thing, you should be able to choose appropriate graphical representation of data. You should be able to choose the right statistical statistical tool. For this, you need to first understand what your data is. It is in what format. Then whether it's a measurement or whether it is a discrete quantitative data in the form of number of people, or whether it is just a nominal data like male, female, so on and so forth. Then you have to first see whether the data follows normal distribution or not. If it does, you go for a parametric test. If it doesn't, then you go for non-parametric versions. This is applicable to unpaired t-test and ANOVA, the table that I had shown you. Correlation, regression. For correlation also, there is a non-parametric version. For regression, you need not go ahead with non-parametric, parametric. But nevertheless, you should do ND analysis. And chi-square test by default is a non-parametric test. So remember, understand the type of question that you are framing. Understand that first. What is your objective in terms of that experiment? You will accordingly then come to know what type of data you need to collect. So remember, statistics is the grammar of science. If I would have just written here statistics, grammar, science, it will not make sense. When I add the right article and the right preposition and right is, it kind of makes sense. Statistics is the grammar of science and research. There is no escaping it. All we can do is embrace it and start liking it. You will develop the taste for it. Okay. So thank you so much for your patience and for kindly listening to me. I appreciate it. Thank you.
Thank you. Thanks, Dr. Shruti. What an excellent message to end the talk, and what an excellent session to bring us those all insights towards biostatistics. We have been seeing through the kind of explanations that you have been giving. It all demonstrated that you have got a lot of passion towards this topic, and we sincerely thank you for taking out time. We have got a couple of requests and posts coming through the chat, so we'll just go one by one. And then we'll ask our audience to go for live questions if they have any. And we'll hand it over to Dr. Kavita for expressing her vote of thanks. So I'll just go to the first request that we have received. Can you suggest any book to brush up basics of statistics?
If I am going to be put on record, so I will tell you two books which I read. There will be many others will be very passionate about it in terms of telling me, no, these books are also there. There are many books. It all depends on the way you understand the language of the book. The first thing is that. So I'll give you names of two books. One is Wayne Daniel, the book by Wayne Daniel on biostatistics. So that is one book. Wayne is Wayne, Daniel. So that is the book that I consider as my Bible in terms of understanding all this. I had referred to it during my PhD, but during the start of my PhD and before that, to understand statistics in terms of the language. Okay, so for some people, understanding the language is sometimes tough in terms of the book, the way it is written. So I suggest go for a simple version, which is I think Methods in Statistics or something by B.K. Mahajan. I would say start with that. Uh, I simply am really being candid here. That is what I have done. I tell my students also to do that because it's a very good book for the initial stages to understand. And then later on, you can go for Wayne Daniel and then read research papers, not research papers on statistics. Read research papers to understand what analysis they have done for their objective. Then you automatically come to know, okay, even I have a similar objective. Maybe I can do this. But the other research paper had the same objective, but they have used another type of statistic. So then you kind of understand the application in terms of what you had read before. And then you kind of understand, okay, these are the two research papers. I think I'll go for this research paper and the way they have gone about it. That's how it is. So I suggest these are the books, Wayne Daniel and Statistics by B.K. Mahajan. These are two books.
Thank you. Thanks, Dr. Shruti. We have also got a request from one of the students to have your email ID. I believe they must be reaching out
Yeah,
to you
definitely.
with their
Why
query.
not?
Yes.
I'm writing it in the chat box itself.
Yes, that
This
will
is
be
my
great.
personal email ID. There's another college email ID, but I suggest you all can send me your queries through personal email ID. I'll definitely help you. It's no doubt about it.
Thank you.
Yeah.
Then there is another question coming is please give us a brief idea about PCA and cluster analysis.
See, that would require another session altogether. I'm so sorry. So, if you don't mind, maybe through email we can have a detailed discussion about it. Whoever has asked this, I hope you understand, because otherwise the endless explanation would be there. But definitely, whoever has asked this, you can contact me through email ID, and I'll definitely enlighten you more about this. No doubt about it.
The question has came from Bhavna Negi. So maybe Bhavna, you can reach out to Dr. Shruti
Yeah,
or
definitely.
an email.
Yeah.
Thank you so much. For our online participants, if you have any questions, please utilize this opportunity and feel free to ask any questions. We'll pause for some time so that we'll give you an opportunity. Okay, if there are none, then maybe Dr. Shruti, I have got something on in mind. If you can just help me get that understanding. As we are practicing lot many things, lot many surveys in our industry, we have been hearing this term statistically significant. So
Yes.
if there is any specific method or number that is to be looked out when we say that okay, this should be a statistically significant, or it really depends upon the depth and gravity of that study. How should we be correlating it when we say that anything has to be statistically significant? So that was one doubt in my mind. If you can please address it.
Okay. So when you said in terms of whether there is any, from what I've understood, you want to know whether there's a criteria to determine whether something is
Right.
statistically significant or not. So there is a standard thumb rule that 5% level of significance or P less than 0.05 is considered to be the critical P value or critical level of significance. So you need to get your P value as small as possible, like I told you. And based on that P value only, you can comment whether your data is statistically significant or highly statistically significant or marginally significant. If your value comes like 0.05, exact 0.05, then you call it marginally or marginally statistical significant or borderline. If it comes something like 0.02, you say there is statistical significance. But what happens is, what we do when we do our kind of analysis, we bring the p values down every time. I mean, we test it at 0.05, 5% level of significance. We test it at 1% level of significance. We try to see whether what is my p value. If it comes less than 0.001, it is highly statistically significant. So
Okay.
meaning remains the same, that you are not seeing some difference by chance. the value is very less, which means it's not by chance, because P defines chance occurrence. The chance occurrence is less than 0.001, which means your data is highly statistically significant. That's how we go about it. Yeah.
Thank you. Thanks for the clarity. So, Dr. Kavita, if you are online, I'll request you to please express the vote of thanks.
Yes. Thank you, Dr. Gaurav. So I have the most important part to play today, which is expressing a deep sense of gratitude towards Dr. Shruti Kakodkar for delivering such a insightful deliberation on biostatistics. I mean, while doing my PhD, I remembered some of the hurdles that I had when I was learning biostats and all. So yes, most of the biologists take it as a very dry topic, but you made it very lucid, very convenient, and very the examples that you were citing at multiple points, I'm sure the students have benefited a lot from there. So I remember once Albert Einstein said that for in every difficult situation there lies opportunity. So I'm sure all the students who are attending this, including all the experts as well, they will make up on this opportunity of including biostatistics in whatever research they are doing, specifically in the food domains. And thank you, Dr. Shruti, for making it up and delivering such a wonderful talk. Thanks also to AFSTI, which has been non-stop functioning and taking initiatives to hold such wonderful programs, getting such wonderful speakers on board, and helping the students rise up their level of understandings. Thanks to the chambers for always partnering with AFSTI and collaborating and making such kind of events a great success. And thanks to the wonderful audience who were there, intrigued by the wonderful presentation and a lot of question answers. Thanks to Dr. Gaurav as well for being there throughout. So thanks to all of you. And I think that marks the end of today's webinar. Thank you so much. Have a nice day, all of you.
Thank you. Thank you.
Have a